So concentration gradients, and what else? There's one other kind of pull that determines where ions go.
Like the gates and stuff? Is that what you're trying to?
No. Has he talked about the electrochemical gradient?
Physics class.
I don't know, I've heard him mention it, but we haven't like
Really talked about it. Okay, so then I'll just kind of keep it simple then. Basically, like you're right, so determines the movement of ions is mainly concentration, but the ions all have charge, right? So when they leave or they move, it changes the charge of the area that they move to or left, right?
Mhm.
So kind of keep that in mind, because that plays a big part in like the charge of the cell, what happens there. So what is the purpose of an action potential? Like, why does our body do it? Why do we even want that to occur?
Just to send signals from what I understand.
Send signals?
Yeah.
Yeah, perfect. Anything else? That kind of cover it.
you mentioned something about like pain levels or something. The better the action potential or something, the better the higher the pain.
Yeah, different frequencies of action potentials determine different things. For like pain, for example, is one. So pretty much, like you said, it's signal transmission, but everything is controlled by action potentials. You know, you move your arm, you move around, you contract muscles. It's all because of action potential sending signals, like you said. So, say we have a nerve here, and it's innervating us. This is just a regular cell. How are you guys with like different gates that opens at which point, and kind of what happens, the whole process?
You mean to let like proteins through and stuff into the cells?
Yeah, well, different protein channels opening at certain times and letting ions through, that kind of thing, and the process. I guess from beginning to end of an action potential, how are you guys with that?
Okay with it.
Second one is just generally just depolarization. All the potassium's leaking out. Depolarization happens with the opening of the sodium voltage gates. And then it repolarizes as the sodium gates close, potassium gates open, and then it'll hyperpolarize for a little bit. Which
What exactly is hyperpolarization? Is it just
You know?
hyperpolarization is just when it goes more negative. Like every membrane has like its resting potential is like generally 70, but sometimes it'll get overflooded because of the active transport system, because there'll be more K's inside than. Or not, yeah.
I don't think I've learned about any of this in my class yet.
You haven't?
I think
Done any action potentials?
the last thing we learned about is the integumentary and skeletal system.
Oh, you guys haven't done that, huh?
No.
Integumentary system. Yeah. Sorry. I mean, it's gonna help, but it's probably not be as relevant for you. So.
Technically, I still have.
You're welcome to stay.
Oh, no, yeah, I'll stay.
Okay. All right. So hyperpolarization. Where would you say that is on this graph? Or have you guys seen this type of graph before? So where would hyperpolarization be?
it'd be kind of like the peak goes up and it goes all the way down and kind of that dip right there.
Right here?
Yeah.
Hyperpolarization.
Yeah.
Yeah, perfect. And then where is depolarization start on this
It
graph?
goes up, like going up to the peak.
Yeah, and then repolarization goes down, right? Okay, so you guys are good with that. So what causes this initial incline towards a more, let's say this is 0, an incline towards a more positive charge?
Is it just like sodium?
Mhm.
Or if it's rushing, I think it's rushing out or in.
Yeah. So, for example, a nerve can release some neurotransmitters. Have you guys talked about ligand gates?
Yeah.
Mm-hmm.
And how do they function? What's their deal?
They have to have like a receptor, like open them for it. They don't just open.
Yep.
I think acetylcholine, right?
Yeah, in a muscle it's acetylcholine, but there can be a lot of different types of neurotransmitters.
Okay.
But for muscle contraction, acetylcholine. So a nerve releases a signal, these could be called a ligand. They bind the ligand-gated receptor, and when they bind, then what happens?
It open.
They open up, right? And that lets in what ion?
Sodium.
Sodium. Awesome. Sodium floods in both of these, and that causes this, right?
Mhm.
What happens once it gets to threshold, which is that dotted line, dashed line?
Mm.
It's like it sends an action potential and all of the, what's it called, all the other sodium gates open up too.
Yeah. What are those gates called? What type gates are they?
That's not forgetting.
Huh?
Is it voltage?
Voltage, yeah. So those work because when the cell gets to a certain voltage, they open, right? Makes sense. So then all of a sudden these guys will kind of swing open, and then lots more of sodium will come in. And that's what causes this steep incline, right? And there's a bunch of them, but calcium voltage gates can also open. So it's influx of positive ions, right?